part 1 of chapter 15, selective breeding. Now, you may have already looked at the other 15-1 video. Um, this is going to be a little bit of a review for you, and there's a little bit of stuff from chapter 14 on this with the some information from the human genome, etc. So what was the goal, or what are the goals, or what is the goal of the Human Genome Project? Well, the big thing as far as the goals of it were to analyze the human DNA sequence. Now remember that the Human Genome Project began in 1990 and involved it was led by the United States and it involved a few other countries. Now in June 2000, they had a working copy of the human genome and it was essentially complete. So it took them about 10 years to identify the genes on there. Now we have research groups that are analyzing the DNA sequence. They're actually figuring out what bases, the order of the bases rather. And biotech companies, which is a new and developing field, are getting information that can help them develop new drugs and treatment for diseases. Because remember when we've talked about human genetic disorders, it's all about the gene, having faulty genes. And if we can fix the genes, then we may be able to help people who have certain diseases. So this data is for the public. And what you can do is that you can go to this website and it'll show you exactly what's going on with the Human Genome Project. Another thing we talked about was selective breeding. And remember, selective breeding was to get the desired characteristics produced to the next generation. So they might mate certain plants or certain animals to get the type of crop that they want, or to get the type of animal that they want. So humans use selective breeding to pass desired traits on to the next generation of organisms. One type of selective breeding is called hybridization. And that's when you get two dissimilar individuals and you're trying to get the best traits of both. Example of a hybrid would be a liger. And we've got a male lion and a female tiger. And a lot of times hybrids are often hardier than either of the parents. Now with selective breeding, another type of selective breeding is called inbreeding. And usually you continue the breeding of individuals with similar characteristics. So remember we had the hybridization was dissimilar and inbreeding will be with similar. And they want to make sure that the characteristics will be preserved. The problem sometimes happens with that is if there's too much inbreeding, and you can get serious genetic problems. So, for instance, white tigers are known to be very, very valuable and very rare. And here's a picture of a tiger that had some genetic problems. Now, another way to manipulate the DNA or to increase variation would be to induce mutations. And there's a couple of ways to induce mutations. Now remember, all mutations aren't necessarily bad. Sometimes, or many times, they're good mutations and they help an organism to survive. So they can increase mutations by using radiation or chemicals. And as I just said, that a lot of times these mutations have desirable characteristics that are not found in the original population. So for instance, here I've got a picture of an orange with seeds, and then I've got one that has had the mutations added into it or induced in it, and then you get a seedless orange. And obviously seedless oranges, are more expensive, they're more desirable than oranges with a bunch of seeds. Another way that they use these mutations is that they developed, scientists have developed bacteria that can help clean up oil spills. So instead of digesting, say, tissue or something like that, these bacteria actually digest oil, and they use that to convert to energy. The big area you'll notice with mutations will be in producing kinds of plants. Now, sometimes these mutations double or triple the amount of chromosomes, and that's known as polyploidy. Remember, haploid was the normal number of chromosomes, diploid was double the number. Well, polyploid is many times that number, and it produces new species of plants. Now, one big thing I want you to always remember is that polyploid in animals is usually fatal. You know, animals cannot have a whole nother set of chromosomes. That's just too much going on. Here's a chart with some examples of polyploid plants that you may or may not know are polyploid. Domestic oat, peanuts, sugarcane, bananas, potatoes, cotton. Okay. The one thing you might want to look at when you're looking at organic organisms, or like say organic strawberries, the polyploid strawberries are huge compared to organic strawberries. And a lot of times that's what happens with fruits and vegetables. If you go to an organic market, those organic fruits and vegetables are a lot smaller. Okay, this is our view of the end of chapter 14 and a little bit of chapter